Good afternoon. My name is Harrison Steedman, and today I'll be talking about a 2024 research paper led by Hicham Tasfayout, exploring a potential therapy for Duchenne muscular dystrophy, also known as DMD. The aim was to observe the effects of split intine-mediated transsplicing of proteins to express large dystrophins in vivo. This area of research is significant as DMD is a severe, progressive, and fatal genetic disorder, and advancements in treatment of this genetic disease may pave the way for treatment of other rare genetic diseases. Duchenne muscular dystrophy is an X-linked disease caused by mutations in the DMD gene, leading to extreme dysfunction or total absence of dystrophin. Dystrophin acts as a sort of shock absorber between actin-myosin filaments and the extracellular matrix. Without it, muscles lose flexibility and are damaged every time they contract, leading to severe issues with posture, mobility, muscle weakness, and repair. As you can see in Duchenne muscle tissue, muscle fibers are disorganized and diminished in size, and with a greater amount of fat and fibrotic tissue. Furthermore, cell nuclei are delocalized, migrating to the center of the muscle fiber, indicating muscle fiber damage. Unfortunately, DMD patients rarely survive into their 20s because of respiratory and cardiac failure, so it's a pretty horrific disease. Current therapies cannot cure the disease, but they do alleviate symptoms and prolong life, so it's the best we have so far. Patients are told to limit muscle exertion and undergo physical therapy. Since muscles are unable to effectively repair themselves without dystrophin, overexertion leads to pseudohypertrophy, where muscle is replaced by fat and fibrotic tissue. Patients are given ventilators and surgeries to open airways, and to improve mobility, they must take non-steroidal medications, undergo surgery on tight joints, and use wheelchairs. However, gene therapy may provide an effective remedy or even a cure. Recent advancements in gene replacement using adeno-associated virus vectors, or AAVs, mean the faulty DMD gene that causes muscular dystrophy can be replaced by a functional gene via AAV therapies. AAV treatment involves replacing the insides of a virus capsid with a desired gene, which then gets injected into various tissues or organs throughout the body to deliver said gene. Unfortunately, this means that the gene must fit inside the capsid, which has a maximum capacity of around 4.7 kilobases, over 500 times smaller than the desired dystrophin gene, with a length of around 2.4 megabases. So how can this gene be squished inside the capsid to be injected into a patient? Attempts to shrink the dystrophin gene by removing coding regions inessential for its function has led to microdystrophin. However, this shortened protein is not as effective as wild-type dystrophin, leading to phenotypes closer resembling Becker muscular dystrophy, with a risk of accelerated lethal cardiac disease. That is when Hicham Tasfayout aimed to find a different solution. And they did. They hypothesized that they could break the dystrophin coding sequence across 2 or 3 AAVs, which they could then inject into the host. These sequences would encode partial dystrophin proteins fused to split inteins, which then bind to their appropriate pair and splice the two protein fragments together, allowing recombination of a larger functional dystrophin within the muscles. It is important to note that this strategy works through protein-level reassembly. Previous attempts at DNA-level reassembly using homologous recombination or DNA transsplicing were often inefficient and relied on the vector genomes joining in the correct orientation. They could also generate unwanted byproducts through vector concatenation, which may cause safety and reliability issues for clinical use. Over 30 split inteins had been characterized as of 2024, and Tasfayout couldn't just use any pair and hope they work together. So they decided to screen 23 of the inteins using a split GFP system to find which pairs had the greatest efficiency and specificity of reassembly. GFP stands for green fluorescent protein, so their system involves splitting up GFP and splicing it back together with different intine pairs inside cells. As the name suggests, GFP fluoresces, so the inteins that resulted in the greatest fluorescence meant that they likely reassembled the most GFP while minimizing the X-teen scar. After 3 screenings, they selected 4 different inteins. They transfected cells with a midi-dystrophin coding sequence via a single plasmid or dual plasmids containing each of the split intine pairs. Western blot analysis revealed strong bands at around 291 kilodaltons, suggesting midi-dystrophin was successfully reconstructed by all 4 intine pairs in vitro. As you can see, the split intine approach resulted in 5 to 8 times higher expression than the single vector approach. Their next goal was to reassemble full-length dystrophin using a triple vector approach. For this approach to work, it would need 2 pairs of inteins that wouldn't cross-splice with each other to ensure orthogonality. So they performed another trial with 2 different combinations of split intine pairs to connect the N-terminal fragment to the middle fragment and the middle fragment to the C-terminal fragment. Western blot analysis showed that both combinations resulted in the expression of a strong band at around 427 kilodaltons, 2 to 4 times higher than the control full-length plasmid. So it is possible to reconstitute full-length dystrophin from 3 separate AAV plasmids in vitro via split inteins. Now they just had to test it in vivo. Unfortunately, this study was limited to mice due to the experimental nature of the therapy, but their genetic proximity to humans mean they make an appropriate model. They injected the tibialis anterior and cardiac muscles of 3-week-old dystrophin null male mice with a saline solution control, single microdystrophin, dual midi-dystrophin, and triple full dystrophin vectors in dosages of 50 billion viral genomes. They used male mice as DMD is an X-linked disease and therefore more common in males. After 5 weeks, they dissected the tibialis and cardiac muscle. Western blot analysis confirmed that the full-length, midi, and microdystrophin were all successfully reconstructed in vitro. They found that the dual and triple vector approaches were much more effective than the single vector approach, restoring around 40% of dystrophin in tibialis anterior myofibers on average. However, they also found that the dystrophin restoration was much higher in cardiac myofibers, which was most likely due to the limited efficacy of AAV6 capsid in skeletal muscles. Consequently, Tasfayat looked to the recently described highly myotropic capsid AAV-myo with hopes that it would increase dystrophin expression in skeletal muscle. They also experimented with systemic delivery through the bloodstream rather than direct intramuscular injection. They administered either a low or high dose into tail veins of 8-week-old dystrophin null mice over a period of 3 months, and it worked. The expression levels of both micro and midi dystrophin were at least twice as high when AAV-myo vectors were administered, and the systemic delivery allowed for smaller dosages despite the longer treatment period. But how does this translate to older mice? Young MDX mice develop only a mild dystrophic phenotype, but deteriorate as they age. 17-month-old dystrophin null mice were treated for 6 months with low-dose AAV9 or AAV-myo vectors to express micro or midi dystrophin. Samples of the tibialis anterior, heart, and diaphragm were then isolated for histological analysis. Unfortunately, all treated groups still had many centrally nucleated muscle fibers, as you can see in this square here, indicating ongoing muscle damage. However, tibialis anterior and cardiac muscles treated with AAV-myo showed modest structural restoration towards wild-type levels, with the greatest improvement from the midi-dystrophin treatment in this right column. These treatments also improved function to near wild-type levels. The results showed that systemic delivery via AAV-myo not only increased dystrophin expression significantly, but it also showed improvements in skeletal muscle contractile force and cardiac muscle pressure development, closer to levels shown in wild-type mice. Unfortunately, these effects were not as pronounced in diaphragm muscles of old mice, but the treatment would still minimize further damage. The balance of evidence suggests that this treatment is both preventative and restorative. This study successfully proved split inteins as an effective solution to the AAV packaging issue. Tasfayat employed comprehensive functional analysis to prove that the treatments resulted in significant physical and functional restoration in both skeletal and cardiac muscle. They measured not only dystrophin expression, but differences such as specific force, cardiac performance, collagen percentage, proportion of centrally nucleated fibers, and muscle fiber area between several different treatments and controls in both young and old mice, making this an extremely thorough investigation into Duchenne muscular dystrophy treatments. Unfortunately, this study was conducted in mice, meaning the therapeutic effects may not fully translate to humans. There are also concerns, particularly with repeated dosing, in regard to immune responses to AAV vectors and the amino acid residues left by the split inteins, which are all of bacterial origins. Further optimization and human clinical testing would likely be needed before this strategy becomes widely applicable. But these limitations may be addressed soon. More split inteins have been identified since the publishing of this article, meaning future studies can explore more options for more efficient reconstitution of proteins in vivo. For example, Zhao et al. Used a CFA intein pair rather than an NRDJ1 pair to reconstitute full-length dystrophin. Another 2024 study, led by Sean Lindley, found that the mRNA transligation at the DNA level rather than the protein level can be used to restore dystrophin, creatine kinase, and centrally nucleated fibers to wild-type levels. To conclude, current therapies alleviate symptoms of DMD and prolong life, but do not cure the disease. Gene therapy via AAV injection is a promising treatment. However, it has been limited by the modest carrying capacity of AAV capsids and high dosages needed. A recent study led by Hicham Tasfayat found a solution to this problem via split intein mediated trans-splicing to reconstitute large dystrophin proteins in vivo. These treatments proved more effective than the current single vector approaches, and further improvements such as systemic delivery via AAV myocapsids led to muscle recovery to near wild-type levels. However, this study was conducted in mice, meaning questions around long-term human safety still remain. Still, this work highlights a promising new platform for delivering large proteins, not only for Duchenne muscular dystrophy, but potentially for many other genetic diseases as well. Thanks for listening.